program. And literally just in December, got her PhD from Texas A&M. And so we're super excited to have her down here to talk about her work. So take it away, Vanessa. So laser go pew pew and forward goes forward. Okay. All right.
All right. Thank you all so much for that introduction. As mentioned, this is going to be a bit about my PhD work, introducing the pathobiome of soil-borne fungal pathogen, the case of Fusarium hyposphere. So we all might already know Fusarium is one of the most destructive plant pathogens to date, as it has hundreds of host-specific groups known as Forma specialis. I like to think that in all these host-specific groups and all these several crops, there's a Fusarium for everybody, and if not, just wait. And so in these host-specific groups that I'll be talking about today would be Fusarium moxiforum, Forma specialis, cubensi, and that is going to be the Fusarium associated with bananas. And I'll be also talking about Fusarium moxiforum, Forma specialis, nipium, which is the Fusarium associated with watermelon. Now, being a fungal pathogen, this is also going to be having a hyphae. So, as you can see, sorry, the video is not working, but these are actually bacteria that are adhering and following the hyphae and the growth. So, these actually is an area known as the hyposphere. This is the microbiome associated with the hyphae, including the biotic and the abiotic, where interactions can occur. This is of great interest in plant pathology because the hyphal tip is used to penetrate and cause disease into the host. Therefore, these bacteria may be associated with underlying functions associated with pathogen success. Now, this is also a great area to study the novel concept of the pathobiome. The pathobiome, again, is the microbiome associated with the pathogen. Again, that's the abiotic and the biotic. Now, in the disease triangle, which has previously been studied to be studying the pathogen and trading into a supple environment, a conducive environment, and this is a full host. Now, we want to go ahead and actually continue this concept a little bit further into a tetrahedral, where we can study the biotic components known as the microbiota and how they may actually integrate with the pathogen, possibly causing synergistically disease within the host. Therefore, to expand on this concept, our objectives are going to be quite a bit to expand on this. The first one is going to be to identify the bacteria around those hyphal tips, differentiating the pathobiome from other microniches, and of course, trying to determine if there's actually some underlying functions that are associated with plant disease success. So to accomplish this, we first have to inoculate our plants using a soil-like media. So this is going to be working specifically with 3 different isolates of FON collected across Texas. We inoculated our plants with our FON2, the adjacent rhizosphere. We then collected our diseased tissues that were going to be right above the soil line, and we actually collected only from that hyphal tip. So again, that hyphal tip is the area, the hyposphere that we are currently interested in studying. Now, I know it looks like we only used 3 generations and 3 plates, but it was actually a little bit more. We were able to run a generational culture-based study using 13 replicates. This allowed us to try to get those that are highly associated with the hyposphere and not necessarily those that were associated with the plant from where they were cultured from. We then amplified those products using 16S RNA. We first did Illumina sequencing with Fusarium multi watermelon and got our OT level results using the mother pipeline, visualized our results in R. But in Fusarium multi bananas, we tried doing something a little bit different. We not only wanted to look at the hyposphere, but we also wanted to differentiate it from other microniches, including that of the healthy and the diseased. So in the diseased plant, we have something known as the dysbiosis. This is the microbiome under distress. So we want to differentiate the microbiome under dysbiosis from that of a pathobiome. So we use nanopore long-read sequencing in-house just to mix things up to see if we get species-level results. And we run it through the epidemy pipeline and visualize our results in R. Now, just to make sure that we did not lose our bacteria, at the different culturing stages, we made sure that the bacteria was still adhering and following that hyphal growth. So this kind of looks like Houston, where we can see there's a lot of traffic in the more sparse areas, but once you get to more active areas, everything is very clustered. So afterwards, we look at the OT other FLSC systems. So we move forward with our nanopore sequencing results at the species level. And again, we're looking at 3 places. We're looking at the hyposphere, which is again is a micro niche, doesn't really have that much area for nutrients, so it's going to be very less diverse in comparison to our tissue samples, which is this is the disease, and this is our healthy. So this in this heat map, we can see that the blue denotes low abundance, and the red denotes really, really high abundance. Now, zooming in, we can actually see that there is a very similar to what we saw in serum watermelon. There is a core. So we can see that Enterobacter, Erwinia, Pentovia are really highly abundant within our hyposphere samples across all replicates. And then here in diseased tissue, we can see it starts ramping up, and there's not really a lot of those pathobionts in the healthy tissue. So this could mean that the Fusarium is actually bringing its pathobionts into the diseased tissue that wouldn't normally be found in healthy tissue. Now, we wanted to actually go into some statistics using a linear discriminant analysis to try to identify if these pathobionts are highly associated with the diseased tissue. In the pink stars, we can see here that those individuals are pathobionts that are significantly associated with diseased tissue, and none were found to be significantly associated with the healthy tissue. Using a NMDS Bray-Curtis analysis, we wanted to differentiate the diversity of microbiota associated with the hyposphere, the microbiome associated with disease, and with healthy. We even then tried to analyze the stochasticity to show randomness from a microbiome losing its functions in the diseased tissue. And we can see here that although it's not significant, there is some stochasticity within the disease. There is a lot more stochasticity in the hyposphere, which may denote that there's less deterministic effects, less functions that are known. And so moving forward, we then moved with a Spearman correlation co-occurrence analysis. So we did this to try to identify if the pathobionts identified have any correlation or any type of assembly within the different microniches that are different depending on the concept of the functions. So all the tags that are identified are pathobionts. Again, these are the organisms highly associated with the disease. And as you can see here in healthy, there's less pathobionts. They're not really in their modules, and they're actually getting along and positively correlating with the rest of the individuals. In the FLC hyposphere, they're all pathobionts, they all get along. But when we get to the diseased tissue, we can see that the pathobionts are all in one hub, and they're actually positively and negatively associating with each other, and they're separate from all these different hubs that are formulating within the diseased tissue. Therefore, this could be the microbiome under dysbiosis, and this is the pathobiome. A way to visualize this in an animation is when there is actually a disease coming into the plant, the microbial system will change hosts. So it'll start recruiting and will start associating with the pathogen and away from the plant. And this is something that I think that we were able to visualize statistically. So overall, this is some wonderful results, but you might be wondering next, well, what is the function? What could be happening? So we threw these onto an ecoplate. So an ecoplate is a 96-well plate with different substrates that we can use to try to understand if there's any type of utilization that's different between the Fusarium by itself at the 490 wavelength and at the 5090 wavelength. And then we have here, we'll see with the bacteria. And as you can see right away, yes, it loves that tween, it loves the glutamic acid and the xylose, but what was actually significant was the malic acid. So malic acid, for some of y'all who already know, this is what plants and other fungi may use to recruit bacteria. And so this is actually showing significance that it is recruiting those bacteria when they are present compared to when there are no bacteria present. So overall, our results found that there is a core microbiome associated with the hyposphere of different Fusarium isolates, regardless of their host type, their soil type. They will recruit and they will keep those bacteria. Furthermore, we have differentiated the pathobiome from a microbiome under dysbiosis or from an uninfected microbiome. We have also emphasized and have shown that the pathobiome is a very interesting concept, but expanded on it in the terms of that pathogens are not invading alone, but they are invading with the microbiome and deserves to be further studied for understanding the pathogen and mitigating disease. But overall, I could not have done this by myself. I had a lot of support from our lab members and from my advisor, Dr. Sanjay Babu, our undergraduates, including Carly, and I would like to thank our funding sources, and of course, y'all today for your time. if you have any questions, I'd be happy to.
We have time for several questions. So, do we have some time? Do we have some questions? Yeah, John Goolsby.
Hey, John.
Vanessa, what program do you use for the Spearman?
Correlation co-occurrence analysis. Thank you so much for asking that. So I use R, but there's a specific package I love to use called MicroEco. I can actually send you the information. I could walk you through it. It's really easy to use. It's something that you can teach to undergraduate and graduate students. Yes, very simple, very easy to use. Hey, Dr. Phil.
So, Vanessa, great presentation. So we have in citrus the same genus, Physarum solanae, that causes dry root rot, a problem that occasionally we see when there is abiotic stress, like drought conditions, waterlogging conditions. So my question is like, how do you intervene that recruitment of pathobiome into that by the fungus? Is there any way that you can intercept that recruitment? Because you said malic acid is very important.
Yes.
What are your thoughts on that?
Okay, so I don't want to give out too much information because our postdoc is currently working on that right now. We're trying to come up with mutants of Fusarium that cannot produce malic acid or other ones that we have already screened out. So Fusarium is actually in a different time, is actually a saprophyte. So we're hoping that by reducing those abilities to recruit, it'll just become a saprophyte, and that will actually be great for the soil. But at this time, malic acid was something that we are looking into, but we are also looking into other mutants of Fusarium to try to reduce that. And that is something that, of course, is somebody else's work. But thank you so much for asking. That is a really good trail of thought. Hey.
Chris?
I love your network analysis. On your analysis, where you were looking at the difference in stochasticity, a couple of slides before that.
Oh yes,
What?
hold on. So sorry. All the animation.
Oh, that's cool information.
Thank you. It's through transition. I recommend using those instead of.
There you go. So what exactly is that stochasticity analysis measuring? Stochasticity of expression can you explain what this
Oh
is?
yeah, of course. So this is something we came up in the lab because again, there's that Anacarena paper where it talks about stochasticity, showing that there should be within a stressed out microbiome. So these are our centroids that we have identified, and all we did was we measured the distance of the centroid from the points. So hypothetically, the further out the points are from the centroid, the more stochastic, the more random, or more different, like lower evenness that we are experiencing. We've moved that to be stochastic.
Okay, so it's almost like the two-dimensional standard error.
Exactly.
Okay,
Yeah.
and why do you think it was so much more in the rhizosphere or the
hyposphere?
Yeah, and everything else.
So this is on a PDA plate, and this is not its normal environment. And of course, there could be also other organisms that are not being shown here that may be what is used to cause there to be more order in the system. But of course, it's a very unnatural system. So this is something that we somewhat expected. Yes. Hopefully through metagenomics, we could actually re-identify the hyposphere, and maybe it wouldn't be so stochastic, but at the same time, there could be other things that we have no idea that could be happening.
Thank you.
Any other questions? Going once, going twice. Sold, Raul.
So there was a restructuring of the bacteria within the microbiome. It was fascinating. What are the triggers there? How does the plant know, hey, I got to do this? The restructuring of the microbiome.
Oh, so I'm so sorry. I'm not a plant person.
Okay.
I mean, that's something again. Yeah, something of interest. We do have our collaborators to thank for taking that on for us.
Okay.
But that is a really good question about how does the plant react to dysbiosis? Does it produce more malic acid? Does that actually help out the Fusarium? These are all very good questions. Thank you so much.
Okay, thank you for that. Okay, last up in our